Welcome back. Our first speaker today is Dr. Lisa Keith. Dr. Keith is a research plant pathologist at the USDA Agricultural Research Service in Hilo. Lisa's research focuses on the biology, detection, and control of emerging pathogens causing disease on crops grown in Hawaii and the Pacific Basin. Lisa integrates laboratory, greenhouse, and field-based research to develop practical disease management methodologies that can be adopted by our growers. Her presentation today is entitled Fungal Attackers of Protea. Dr. Keith, thank you for joining us.
Thank you, Glenn, and also thank you to HFNA for this opportunity to share our research today. Before I begin, I really want to acknowledge my amazing lab crew, Lionel, Ava, Karma, Brian, Caitlin, and the rest of our amazing team with this research, Hannah, Havila, Pam, Roxana, and Kathy. To get started today, this is actually what I'll be talking about, the talk overview. Some of the general concepts and definitions of plant pathology, like how is disease caused, and what is a disease cycle, and how do you prove pathogenicity. Then we'll move on to the main subject of the fungal attackers of Protea. I'll give a quick update on some emerging pathogens on some other crops in Hawaii and some to look out for, and end with a summary and some overall conclusions. So to begin, plant disease can actually be caused by infectious or biotic or microbial agents, and it can also be caused by abiotic or non-infectious agents like chemicals, the weather, mechanical, even nutritional deficiencies. Disease is actually a process or a change that occurs over time. So it is not one day, nothing, and immediately, like a lightning strike, you see something. That's not considered a disease. But in order to understand what a plant disease is, you have to understand what is normal. The color change you're seeing, or the death of tissue, or wilting, some kind of defoliation, is that normal, or are those the symptoms of a plant disease? And you're going to hear a lot today about symptoms versus signs. The symptom can be defined as the host response to pathogen attacked. So you're actually going to see a detectable change in either the color, the shape, or the function. It can actually be localized, maybe a spot on a leaf, versus systemic that works its way through the vascular system. Sometimes symptoms can be macroscopic or visible to the eye. Sometimes, especially during the early part of disease, it might be microscopic symptoms. Now, that's in contrast to the signs or the actual physical presence of the causal agent. So now you're going to actually observe maybe the fungus or the bacteria present. Now, it's critical when we talk about plant pathology and plant disease, you have to prove pathogenicity using a number of detailed steps before you can conclude that that pathogen is causing a disease. And it's also tricky sometimes when pathogens are considered primary or secondary or opportunistic pathogens, and we'll kind of get into that. Okay, so how is disease caused in plants? Every plant pathologist refers to the disease triangle, and it seems pretty simple, but unfortunately it's not. There are three main components to the disease triangle. You have to have a susceptible host, you have to have the pathogen, and you have to have a favorable environment. And all parts of the triangle have to occur at the right time and the right place in order for disease to occur. So if there's any way to minimize or eliminate a portion of this triangle, you can begin to manage disease. Now, I said it seems simple, 3 parts of a triangle. I thought I'd go into a little more depth for each one. The pathogen, Well, the main pathogens are fungi, bacteria, viruses, and nematodes. And that's actually the pictures you're looking at on the screen here, starting in the upper left. Many microorganisms exist. Thank goodness, most of them or many of them are beneficial, and actually only a small portion of them infect plants. So there's actually very few plant pathogens, even though they can do major damage. Okay, so the fungi, some of you maybe are more familiar with bread molds or mushrooms. These are a type of fungi. The pictures on the right are an actual pathogen growing on a plate, and the spores of that pathogen you're looking at under the microscope. So fungi are actually the most numerous and economically important class of plant pathogens. They have complex life cycles, and there's a lot of diversity with fungi. Infection for plants, when it comes to fungi, not only do they infect through natural openings and wounds, but the main difference between this pathogen and other pathogens, they can often directly penetrate the host. Okay? They produce spores that can be dispersed by wind and water, even humans or animals, and they're oftentimes identified by their morphology, their reproductive structures, or even by molecular means. Okay? Bacteria, they're more primitive than fungi, a more simple life cycle. They exist as a single cell. A mother cell produces more daughter cells, reproducing by binary fission. And they infect plants either through natural openings or wounds. Okay? They cannot directly penetrate the plants. Oftentimes, bacteria can survive on the surface of the leaf. That's known as epiphytic growth. And a telltale sign is if you take some of that infected material, you'll see bacterial oozing and streaming, which is actually what you're looking at the top right. Okay, viruses and viroids are a whole nother class of pathogens, extremely tiny. They're protein and genetic material. They insert into the living host by some kind of vector, usually some kind of insect. They can be transmitted through seeds and tubers, grafting, even physical contact, so it's important for management purposes to know this. And oftentimes viral diseases have kind of the telltale characteristic symptoms, and then you can also ID them by microscopy and other serological tests. Okay, nematodes are round, thread-like worms. The plant parasitic nematodes actually have a specialized mouth part called a stylet. They live in the soil, they feed in and on in plant roots, and we are very fortunate to have Dr. Roxana Myers as our resident nematologist who will answer all of your questions. Okay, so that's one part of the triangle. The other part, the second part's the host, okay, and it's not just resistant or susceptible. There's actually many levels. Sometimes there's a hypersensitive response where the plant can defend off pathogen attack. Sometimes that's invisible, sometimes that's visible. Now, healthy plants can be attacked by primary pathogens. Sometimes under stressful conditions or an unfavorable environment, it kind of sets up the host to be invaded by other pathogens like secondary pathogens. Okay? Sometimes a resistance host, the pathogen can actually overcome that resistance, and more often than not, monoculture can lead to major issues in a field. Okay, then the third part is the environment. It seems pretty simple, but there's a lot of parts to the environment, being the air and soil temperature, the amount of rainfall, the percent relative humidity, even the light can affect spore viability. So there's a lot going on with the environment. Okay, so we'll move on to the disease cycle. In order for disease to occur, well, first of all, there has to be some kind of inoculum. It has to get to the plant host and actually get inside and start growing within the plant. Okay? Once it starts to colonize, it reproduces. You start to see symptoms. It produces more inoculum, which can then get to more healthy plants, starting the disease cycle again. And oftentimes, there's some kind of pathogen survival between the growing seasons, even if there's the absence of the host. Maybe there's an alternate host, like a weed, that the pathogens can survive on. Now, I mentioned proving pathogenicity. Just because you find one bacteria once doesn't necessarily mean that is a true pathogen of a specific plant. Now, Koch put forward these 4 steps in the late 1800s for some animal diseases, actually, and plant pathologists have also adopted this. The 4 steps being the pathogen always has to be associated with all diseased plants of certain symptomology. Okay? You then isolate the pathogen, you grow it in pure culture, you identify it, you then inoculate the pathogen into a healthy plant, reproducing the exact same symptoms that you see in the field. The last step being you've now re-isolated the same pathogen you've put in, thereby proving pathogenicity or proving Koch's postulates. Okay, so to mention disease control, unfortunately, plant diseases are very difficult to control. It is key to be able to recognize the disease you're looking at. You have to have reliable methods of detection, and I mentioned that could be through morphology, looking on a plate under the microscope, or using some serological or molecular techniques. There are many ways to actually eliminate pathogens. You definitely want to maintain pathogen-free plants and distribute pathogen-free plants. More often than not, it takes an integrated pest management system to reduce disease to an acceptable level. We always hope we can eradicate a new pathogen, but more often than not, once it is in a new place, it's very difficult to eliminate 100%. There are management practices listed here: evasion, exclusion, even resistance in therapy. If you want to know more information about that, I'm happy to give more info. Okay, so kind of the meat of the talk, Protea. Fungal attackers revealed. This is our large collaborative and cooperative project with our amazing team. The Protea on Maui is an amazing genetic resource, collected and housed, and they've been suffering over the years. We as a team are really trying to rescue and better understand the host pathogen interactions, the accurate diagnosis, so we know how best to manage what is going on there. So the folks on Maui actually survey for the disease symptoms. They collect the samples and send them to our lab at PBARC. We're both cataloging the Protea diseases. We then photograph and describe the symptoms and the signs we see. Remember, the symptoms, how the host responds to attack, and any signs. Do we see the physical presence of a fungus? Okay? We then isolate and purify these suspect fungal pathogens, identify them by morphology and molecular techniques, store them in a permanent culture in case we want to refer back to them, and also keep them actively growing in the lab in order to actually do tests with them, either pathogenicity tests, fungicide tests, and the like. Okay, so again, the credit to the lab crew. More than 70 samples not only were collected, but this is just a snippet in the last year of, well, the great description of the plant and then where the sample has come from, and you can see listed all parts of the plant, above ground and below ground. The type of symptoms observed are described, and then through morphology and or molecular identification, we've been able to essentially ID the fungi, the genus, and most species. Okay, so as far as the symptoms go, unfortunately, some of these main disease symptoms, I wish I could say there really was just one major leaf spot. Unfortunately, that's not the case. There are instances of leaf chlorosis or yellowing, leaf necrosis or browning of the leaves. There are various leaf spots. There's marginal necrosis. Sometimes there's tip dieback of branches or tip necrosis of the leaves. We've seen symptoms of branch dieback, various stem cankers, problems on the main trunk and the lateral stems. When you actually start looking inside the plant, we can see discolored pith or the cambium. There's some nodal discoloring, and there's even discolored roots. So, does that sound complicated? It is. Fungal suspects. So we're always hoping to find maybe that one or two primary pathogen causing the problem on these plants. We have found more than 20 different genera associated with these symptoms that have been isolated, purified, and identified. Now, I'm not going to go over all 20. Some of the most common fungi have been Neophysococcum, Pestalozziopsis, Phomopsis, Alternaria, and Fusarium. Okay, so I thought I would just show some pictures of these major genera just to get a better understanding of the picture of these symptoms caused by these main fungi. So with Neophysococcum, more often than not, these leaf spots exist and expand. They start very small. They increase in size, kind of an irregular, circular manner, with dark necrosis. As it gets larger, the center turns even more brown, and these spots expand and start to take over the leaf material. We've also seen instances of kind of this tip necrosis on leaves, and that's what you're looking at in the top right. Okay, so Neophysococcum also causes stem canker, which is the left two pictures. The top actually shows that part of the main plant seems to just die, and oftentimes rapidly. If you zoom into the areas, you can very clearly see a stem canker. And then the other two pictures are the example of pith discoloration. Okay, Pestalozziopsis, characteristic tip dieback, also causes stem cankers and leaf spots. Okay, Phomopsis causes a stem canker. As you can see, a lot of the stem cankers look alike. Alternaria causes a stem canker, and also a variety of leaf spots depending on the host material. Okay. Fusarium was isolated from below ground, from the roots, the soil area, causing kind of a rapid wilt. Okay. Now we knew what major pathogens we were dealing with. How do you manage them? Well, we started looking at fungicides in the laboratory. Here's a quick picture of an in vitro assay to test fungicides. You remove a portion of the media, you replace it with actively growing fungal culture. The media is amended with fungicides. After 7 days, you measure growth compared to a control that doesn't contain fungicide in the media. Okay? The left would be no fungicide present, 2 different isolates. The middle's what you're kind of looking for, minimal growth, even better if there's no growth present. Here's the list of 7 different fungicides that were looked at with various active ingredients, various resistance or FRAC groups, which is really important to pay attention to when it comes to rotations, the various label rates, and the tested rates. Okay? We looked at 10 different isolates of the main genera that I mentioned earlier, which came from a variety of hosts and caused a number of symptoms. Okay, and I'm going to say this is preliminary lab results. This is the effectiveness of these fungicides based on inhibition of growth. And we can now see, although work needs to be done a bit more, kind of the top couple fungicides to inhibit the growth of these major pathogens. But I'm going to mention that so far, really, we only see complete inhibition with Alternaria. So while we really do decrease the fungi's ability to grow, right now it is not stopping growth completely. Okay, so when it comes to management of these fungi, definitely it's a lot of what you can do in the field to increase air circulation and eliminate water on the tissue. Yes, fungicides sometimes as a preventative. Most of them will not cure once they become infected. Elimination due to sanitation, proper sanitation, getting rid of those infective plant parts in the field. Thinking about, well, are there more resistant varieties to plant? And also still needing to really sort out, are we talking about primary or secondary pathogens? Is it because the plants are a bit stressed in this unfavorable environment that these pathogens are now kind of taking advantage of that situation, which a lot of these that I've mentioned are. They're either present on a lot of other plants or could be present inside as an endophyte. And once the plant gets stressed, now these fungi decide to do what they can do. But that wouldn't occur with completely healthy plants as a primary pathogen. Okay, I know I'm real close to the end of time, so I'm just going to fly through this part. Just mention a few emerging diseases on other crops just to remind you what's here and also to pay attention to perhaps what could be coming. So Rapidohea death caused by a ceratocystis, 2 new ceratocystis species, needs a wound to get in. Okay, but if even the most minor wound on the most healthy tree, once these ceratocystis species get in, the result is death. And there's maps to where these pathogens are located. Okay, the newest invasive species is coffee leaf rust, the obligate parasitic fungus called Himalaya vistatrix. Now, with rust, the abundance production of spores, the ease of distribution, first being detected on Maui, the end of October 2020, within 6 months, is now present on all coffee growing areas of our state. These are some early symptoms, the chlorotic spots, then the spores, which is actually the sign of the pathogen. You can see as it gets bigger, necrosis follows, and ultimately leaves start to defoliate. Okay, a couple things to kind of pay attention to that are not here in our state. There's some new cacao viruses that should be on our radar, and these characteristic symptoms of either white vein clearing, red vein banding, they are vectored by mealybugs. Okay, that's kind of the key. And a second one being laurel wilt. Really causing a major problem on Laraceae, on the mainland, but the importance is avocado is part of that family, okay? It's not a preferred host, but these are some of the symptoms. Oftentimes a very rapid death of the plant. The avocado leaves will remain attached for months, looking brown and necrotic. If you look at the sapwood, you're going to see internal discoloration, and the presence of beetles is a telltale sign, and that's the boring dust that you're looking at. Okay, some overall conclusions for the talk. Prevention rather than cure. Okay, it has to be a holistic approach. The best way to delay the onset of symptoms, either reduce the initial levels of disease or reduce the rate of the disease development, is key. Okay? You have to have kind of a short-term and long-term solution. It is critical to accurately determine what pathogen you are working with in order to develop the best and effective management techniques. Okay? The awareness of the existing diseases is critical, which is really what we were trying to see with Protea moving forward. Now, prompt diagnosis of new symptoms or unrecognized problems is also critical, could be something new. Management definitely depends on the accurate understanding of the pathogen you're dealing with and how those different diseases will impact production. Now, unfortunately, in this day and age, new diseases will emerge due to international travel, the changing environment, perhaps change in production practices, but we're here to say, please do not pack a pest. And with that, I say thank you so much, and I'm happy to answer any questions.
Thank you, Lisa, for that presentation. And I'm going to tell you, we have been swamped with all these questions. So the first one is, so the cacao diseases that you have discussed, they haven't been found yet in Hawaii.
That is correct. We have not found cacao viruses that were presented here.
Very good. Okay, why is it important to get rid of diseased plants in the field?
Okay, great question. So those diseased plants contain the pathogen. And if you just leave them, it can serve as an inoculum source to start new diseases. So it is critical to remove, by good sanitation, that inoculum source.
Okay. In going back to your Protea discussion, were any of the Protea diseases a surprise to you?
Great question. And actually, no. So that's the good news. No. Most of the genera are typically found on Protea growing around the world, the types of leaf spots, some minor canker issues. Most actually are not considered primary pathogens. So I think that's good news moving forward.
Okay. So, I mean, is there something, you folks are always on the lookout for a possible disease that is found elsewhere in the world that may come into Hawaii at some point?
Yes, it's always good to be well informed of what is out there and what potentially can come in. But I'd say even more critical, you as growers know best what your plants look like, and if something doesn't look quite right, the best thing you can do is get a sample in to see if it's something new as far as a pathogen.
Is there a potential to reduce root pathogens by using anaerobic systems to compete? And how long does that process take?
That's a great question. That probably will take far more than the time allotted. There are a lot of potential management systems, that being one. I can't give any more detail than that, but we'll definitely be looking up that information, and please feel free to contact me. My email's on there. Happy to answer any more questions that you may have.
Okay, the final one. If someone has a disease sample, do they bring it to you?
Well, the best answer to that is please contact me first. You might have noticed, the amazing diagnostician Brian Bush is now working part-time out of our lab. We're very fortunate to have him. So please do not just send every and any sample to our ARS lab, but definitely contact me and we can help sort it out.
Thank you very much for your presentation.
Thank you very much.